The joy of learning in a professional course or a skill development program is only when you get placed in a company, in an industry, correct? And I'm sure many of you will agree with this fact. But many a times you pursue your program and no placement happens. But if you remember, 3 months ago, Biotecnika, in collaboration with Quantumzyme, came up with a placement guarantee bioinformatics training program. And today, we are right here in Quantumzyme after 2 months of the start of this program. So just to remind you, we started this program on 1st of February with Quantumzyme. So our students were placed, so they were trained in the entire January, and in February they were placed in Quantumzyme. So today we are here in Quantumzyme and we are going to interact with those 5 students whom we have placed in Quantumzyme and to know their experiences, what they have learned in the last 2 months, and we are trying to understand that whatever we train them on bioinformatics, have they applied it in real world, in the industry, and what is their experience? So let's go and find out what is their experience, how are they learning, how is their career progressing once they finish the program of bioinformatics in Biotecnika and they got placed in Quantumzyme. So let's go inside and find out what's going on. Welcome back. So, as I told you earlier, in January of 2024, we had launched the Bioinformatics Quantumzyme internship, wherein we were providing guaranteed placements to our interns. And today as I told you, we are in Quantumzyme, we are in the conference room, and in front of me, we have the members who were placed by Biotecnika in Quantumzyme. So we have Prasna, we have Glenn, we have Durj, and we have Jyoti. So hi guys, how are you? Good? Wonderful. So these 4 people, in fact 5 people we placed in Quantumzyme. One girl is not well, so she's not here. But apart from that, we are here today to showcase what exactly they have done in the last 2 months. So remember, this internship is for 6 months. So they are already placed here. They're already working on novel enzymes and various other things. They are working on bioinformatics projects. So they are going to present, they're going to tell us what exactly they have done in the last 2 months, so that it will serve as an inspiration to all of you, so that you know, okay, when you join the industry, what all things you do, what all things you will be able to do, and what the industrial setup means. Okay. Now, at this point in time in this video, you must have a lot of questions. So feel free to put them down while I'll ask my interns from Quantumzyme to come over and start presenting. Over to you.
Hi everyone, my name is Prasna. I'm going to be presenting my internship report. I've been working at Quantumzyme as intern for 2 months. So as we can see from the skills that I've gained, the progress, literature search, sequence alignment, ligand editing, searching, Swiss model, and docking visualization, MD simulation, and MD simulation, ligand parameterization. So these skills enabled me to work on the collection of suitable mechanisms for the catalysis of enzymes. Then I was able to learn about the databases, BLAST, JALView, Cluster Omega, and I was able to do ligand editing, means I can edit chemical structures in JavaScript format. Online, and I can create Mol2 files and give them as input to study the docking for enzyme ligand docking process. And then moving on, there were many visualization and analysis tools as well that I learned, such as VMD, Chimera, Pymon. Then I was able to learn GROMAX for MD simulation, and then CGNF for ligand parameterization. This is my progress for Feb month. So I've done literature search for keto reductase and asthma drug intermediate. and I've done the patent search for that as well. These are the databases that I've learned: Brenda, NCBI, PubChem, and RCSB. So Brenda enabled me to search enzymes based on like the substrate interaction, the pH, temperature, and various other factors. And then NCBI enabled me to look for the literature as well as the protein and genome information and all. And then PubChem, I used to get the structure of the ligand molecules. And RCSB is, like, as you know, it's a repository for protein PDB structures, which we used to do docking and all. So going for the alignment tools, this is the BLAST tool. So I've done an example search for 6EQE. And this is the Clustal Omega, and this is JALView. So BLAST and Clustal Omega are like multiple sequence alignment tools. Then JALView, using JALView, we can compare multiple sequences and analyze the data and all. So I've learned Swiss model, protein modeling tool as well. If we give the protein sequence, it will give us a structure. It will completely model, and it's an online based server. You can download the data from there. So as for drawing tools for ligand identification, I used ChemSpider and Avogadro. So these are the docking tools that I used: Autodoc Vena and Autodoc 4. Autodoc Vena gives us the result in terms of binding affinity, whereas Autodoc 4 gives us the result in terms of binding energies. This is the command tool that I used, Mobaxterm, to run the scripts and the docking processes and all. As for the visualization tools, this is Chimera, this is VMD, and this is PyMol. I just generated my docking process and visualized them and analyzed them in these three visualization tools. So moving on, I did docking analysis for the protein called 6EQE. It is a polymer, PET polymer degrading enzyme, and we used like a PET sample that I drew from the chemical drawing tools that I mentioned earlier. And as you can see, these are the binding affinities, and those are the docking process, like how it's interacting with the protein. Moving on to keto reductase candidate. This is keto reductase obtained from Candida albicans species, and then the same process what was done for that as well, docking and the binding energies and the distance between the interacting substrate and enzyme atoms and all. So for the month of February, we were given an assessment. We had to find out which out of the three enzymes were suitable for catalyzing an anti-diabetic drug. So on the top we can see the substrate, and below we can see the product. These are the enzyme sequences that was given to us, and we should we modeled them and saw the catalytic process. So these are the mechanisms of reaction for those particular enzymes. And as for my assessment methodology, I did the sequence search using BLAST. Then I did Swiss model to obtain the protein structures. Then I edited the structure using Chimera, and then did both Autodoc 4 and Autodoc Vena rigid docking. And then I visualized the distance using PyMod. As for the docking analysis results, for enzyme 1, we can see that the distance is in Autodoc 4 is 3.375 and 3.0 in Vena. For enzyme 2, AD4 gave us 5.6 and Vena gave us 3.1. For enzyme 3, we got 3.5 distance and 2.9 distance for Autodoc and Vena respectively. So the basis of this is that the lower the distance, the better the catalysis. So based on that, these were my interpretation and results. So the first enzyme is 5FR9, which is according to my analysis, proven to be the best candidate for digesting that substrate into a product. Okay, moving on to the progress of March. In March month, we learned ligand parameterization, for which we used CGenF. CGenF is a tool which adds on charm phosphates to our ligand molecules. So this is the process for generating CGNF based ligand output files. So we can use that output file for MD simulation run, etc. And moving on to modeler. This is the process of modeler where we can input a sequence, then we get a structure out of the given sequence. The modeler program compares our sequence to a bunch of templates, and then it recognizes the structure out of like conserved regions and all. Then it plots a dendrogram, and then it chooses the best protein as a template, and then it models the structure based on that. So as we can see from this graph, we have created a successful model because the alignment position of model and template are matching very well. The red is the model and green is the template. And moving on to GROMACS MD simulation. This GROMACS MD simulation is where we create proteins and ligands in force fields and in simulation boxes. This is an attempt to see what would happen in real life if such enzyme ligand interaction happens. So it is a very powerful molecular dynamics tool, and it's widely used for computational chemistry and biophysics. Then it enables simulation of nucleic acids and lipid membranes as well, not only protein ligand. Then for the analysis part, I've taken an example to show where the ligand and the RMSD of protein backbone are analyzed. So moving on to the ongoing work, I'm currently doing 6EQE ligand, MD trajectory analysis, and PC analysis for MD simulation data, design and calculation for MD simulation box, and I'm also learning various concepts of MD simulation, its analysis, and there are various literature survey and discussions related to that as well. These are my future work: Bash Python, SolidProt, QRDoc, GROMACS VMD TCL script, trajectory analysis, and plotting. Thank you.
Hello, my name is Glenn. This is my intern update. I am working here from 2 months. So let's begin. These are the contents. These are the things I have done here. So this is my February report. First, what I did was literature search and literature and patent search. So before moving on to a project, first we need to find a research task. So for that we need to find various things. So for that we do literature search. Here we were given a task to find biocatalysis between a drug intermediate. So we had to search various databases such as PubChem, Google Scholar, and NCBI. Also, while going on through the project, we discovered that some patent was filed over that protein. So we went and searched that protein, and we got the PDB ID for that protein. Next was the ligand structure building. So after we get the protein, we need to build the substrate. For that, we have various tools such as Avogadro, Marvin Sketch. So what we did was we built the structure using Avogadro. So in Avogadro, you have various things. You can input elements and other bonds, and you can build it. So next, after building this protein and after getting PDB from RCSB database and also building the substrate, we need to visualize this thing. So we have various software such as Chimera, PyMon, and VMD. So in these softwares, we can visualize the molecules. We can see how the protein is, we can measure the distance between the substrate and the protein, etc. So next, after visualizing and checking everything is proper, we will do sequence and multiple sequence alignment. So for sequence search and multiple sequence alignment, we have tools such as JALView, Cluster Omega, and also NCBI Blast. After that, after finding these things, we will move on to docking. So in docking, there are two types: rigid docking and flexible docking. So this docking can be done through two softwares. One is Autodock Vina and other one is Autodock 4. So in Autodoc Vina and Autodoc O, what happens is when we we have to process the protein. Also, we have to process the substrate. Then we input it to the software. The software will calculate all the parameters and it will simulate the docking process, and it will give us the result. After getting this result, we have to visualize it in visualizing softwares, and also we have to see if the substrate is going near the protein. So after that, after learning all these softwares, we were given an assignment. So I will talk about our assignment. So we were given 3 enzymes. So we had to find which enzyme has the best affinity towards a particular substrate. So this was the flow chart. We had to identify the substrate and product. We had to do blast search. After that, we had to obtain the PDB ID. After that, we had to do literature search to find active site and also the reaction mechanism. After that, we were given a protein sequence. So from this protein sequence, we had to find which PDB ID and get the PDB structure. So after getting all the things, we had to do docking. So this was the reaction mechanism, and these were the blast search. We got these PDB IDs. After that, we did sequence alignment to see is there any similarity between the proteins. These were the protein structures we got from the RCSB. This is the through literature search, we came to know various things such as cofactor is present or not, what are the active sites, what are the inhibitors, etc. So these are the docking results. These are the values we get through Autodoc Vina and Autodoc 4. After everything, our conclusion was that Autodoc Vina gave us that enzyme 1 is having more affinity, and Autodoc 4 gives us that enzyme 2 has best affinity. So we can move forward with this. So this was my March update. After the first month, as we went through the second month, we learned modeling and MD simulation. So modeling we can use mainly 2 programs. One is Modeler, another one is Swiss Model. What happens is sometimes protein structure is not available. Only we have sequence. What these programs do is they take the sequence and they build the protein so that we can use for docking. So first one is Modeler software. Second one is Swiss Model. So Swiss Model is various other tools are available. Swiss Model is an online tool. It is more convenient. We just have to give the sequence. It will search for template. Then we can just tell it to build the model. It will give us the protein. So next one is Modeler. Modeler is it is a command-based tool. So you don't have a graphical interface such as we use normally. So everything is done through coding. So, but advantage here is it is lot more flexible than other tools. Next is we'll move on to molecular dynamic simulation. So in molecular dynamic simulation, there is a tool called GROMACS. So GROMACS is mostly used for molecular dynamic simulation. So what we do here is in nature, we got the docking process. After docking, we'll get a complex. We'll take that complex and simulate in GROMACS. So what GROMACS does is it will simulate the environment around the protein and the substrate to see that what happens if water is there and other ions are present. So normally in reaction in the industry or anywhere, they put various solvents and such. So in docking, we can't do that. To simulate that step, we have to use GROMACS. So we can do various RMSD analysis and RMF analysis to see if it is working or not. If it is not working, we have to go back again and redo the docking and other things. So skill that I have gained. These are the skills I have gained. This is the process of protein engineering. First is literature search. Literature search through various things such as PubChem, Google Scholar, NCBI, Google Patent Search, etc. After we do get a grasp of theory, we have to get the structure molecule. So for that, we have Avogadro for ligands and also RCSB PDB database to get the protein. Next, we have to visualize this protein to see that if it is if everything is proper or not. After that, after checking everything, we have to go to docking. Docking, we have various tools such as Autodock 4, Autodock Vina. In these softwares, docking is done. Further, if we get that PDB ID is faulty and structure is not proper, we can build the molecule using Swiss modeler. So these tools like modeler and Swiss model, it will help us model the protein. Next, we have GROMACS. So after building everything, last step is simulating. So we will simulate how this protein acts in nature using this software. So this is how in silico protein engineering is done. So this is the ongoing work we are doing. So PC analysis and network path analysis. These are the analysis that are present in GROMACS. So we are going to learn how it works. Also, we are going to learn about GROMACS theory, how does it work, and what is the backend process. Also, these are the skills I have gained. Literature search, PyMol, Chimera, and VMD, BLAST, JALView, Cluster Omega, Autodock tools such as Autodock 4, Autodock Vina, MOBAX term, and also molecular building software such as Avogadro, Marvin Sketch, protein modeling, modeler, Swiss model, MD simulation, software such as GROMACS. So in the future, what I am going to learn is Bash and Python, Soliprot, KVerdoc, GROMACS, VMD, TCL scripting, also trajectory analysis using GROMACS and other things. Thank you.
Hello everyone, my name is Durj and I have been working as an intern at Quantumzyme for the past 2 months, and this is my internship update. So yeah, starting with the content slide, as you can see, this is what I have done. It's like the full list of the things. So then moving on to the timeline. So what I did was first starting with the software installation and basics, then moving on to literature and patent search, and then moving on to the molecular docking software, and at last for the month of February, I did multiple sequence alignment. So what all software I installed and like what are these softwares? I'll be listing it all in the upcoming slides. So yeah, I started first with Marvin Sketch, which helped me to draw structures of ligands and all. And then the Avogadro, Chimera, PyMol, VMD. These all are like visualization software which we can visualize the protein and the structures, and we can edit and do lots of other stuff. Moving on to Autodock tools, where we actually make the protein, like make the PDB files into PDB-QT files, which we'll be using further in docking process. Same goes for the ligand. And then moving on to Autodock. So there are 2 types of software for doing docking, but there are many others, but I did this 2. I use this 2 software. First one is Autodock 4 and the second one is Autodock Vina. And now if you if you have the question like which one should we use, we should I would say we should use Vina because it's much more consistent than Autodock 4. And now the BLAST, which we all know as like basic local alignment search tool, it helps us search protein structures with the sequences that we have. And moving on to multiple sequence alignment software, that is JALView and Cluster Omega. This JALView is software that we use, and Cluster Omega is like a free software, like it's not which is available in the internet. So MOBAX term is like a Linux environment where we can like do stuff using Linux commands and all. Moving on to literature and patent search. So I was given the task to perform enzyme discovery for the conversion of an asthma drug. So this is the flow chart, like how I did my literature search. And at last, I was able to find the PDB ID 4RF2. Now, moving on to molecular docking. So there are 2 types of molecular docking, rigid and flexible. And I did both of those, rigid docking and flexible docking, using both Autodock 4 and Vina. So this is just the flow chart of how the process goes on. And and like Autodoc 4 gives us the binding energy value, and this is the same flowchart for Vina, and Autodoc Vina gives us the binding affinity value. So moving on to multiple sequence alignment. Just for example purpose, I did this multiple sequence alignment using two PDB IDs that are 6CQE and 7OSV. So which gave me the percentage identity between those two proteins. And now at the end of month of February, I was given this assignment where I had to identify 3 enzymes, and out of them, like which has the best binding affinity towards a drug. So this is the normal flow chart of how I proceeded. So these are the 3 proteins that I found out using BLAST, and then I did the docking, which you can see, and this is the like binding values of binding energy using Autodoc 4, and these are the pictures of the when I visualized it in Autodoc tools, and then moving on to Vina, using Autodoc Vina, I found out this same using the same enzymes and ligand, these binding affinity values along with the distance, which I further visualized in using VMD, which is also a visualization tool. So coming to the conclusion, I got these results for Autodoc 4 and Vina, and as you can see, the best binding energy was the first protein in using Autodoc 4, and using the third protein, I got the best binding affinity values for Vina. So now moving on to March, update for March. So this is my timeline for March. First I did protein modeling, then ligand parameterization, and then molecular dynamic simulations. So what is protein modeling, you ask? Protein modeling is just it's a software, we use 2 software basically. One is Swiss model and another is modular. So Swiss model is absolutely free and it's available in internet. So what we did was if there are protein structures are not available, we just have the sequence, or if it's available and it's there are some fixes to be done in that protein file, we can use Swiss model or modular to do the same. And using modular, you can also add mutations or add cofactors in the modeling process so that we get a different protein model at last. Next, moving on to ligand parameterization. So I used only CGenFF for ligand parameterization. So what it does is it uses the charm program, charm force field to make the ligand file. And since we can't do ligand parameterization using GROMACS, which is used for MD simulation, so we use this CGenFF tool. Last but not the least, what I did was molecular dynamic simulation using GROMACS. So I did for the protein, only the protein, and also the protein ligand complex. So molecular dynamic simulation is simulating a protein, after docking, what we get a protein ligand complex, and simulating it in conditions where conditions of nature and all. So we can know how the protein functions, what all angles it forms, what are the bonds available in the proteins, and how the atoms move and stuff like that. So what we did was at last we found out the RMSD value for the protein ligand complex, which came out to be pretty good, since you can see in almost a straight line. So yeah, this is the work in progress. So the first is PCI analysis for MD simulation. Second is trajectory analysis. Third is literature search, which we are still doing for certain projects and all. The final is learning advanced concepts of MD simulation. And these are the skills that I learned and the skills that I have to learn, which are Bash, Python, some advanced concepts of MD simulations and all. That's all. Thank you.
Hi everyone, this is Jyoti Rana and this is my internship update in the Quantumzyme. And this is the learning path I have done in my last 2 months. The first is literature patent search, Marvin sketch Avogadro, Autodoc 4 and Autodoc Wiener, Chimera, Pymol VMD, with Gelview, Blast, Brenda and Cluster Omega. Then the assessment and modular Gromex and CGNFF. And this is the progress in my February month. They are given the objective. This is the objective they are given to me, and I have to find the literature search for that objective. After that, I did the Marvin sketch and Avogadro for the building a structure of a PET molecule in 2D structure of PET molecule in Marvin sketch and 3D structure of PET molecule in Avogadro software. Then I did the visualization. I did the Chimera, VMD and Pymol for the visualization, and these visualization tools are also used for the further results of docking and also for the MD simulation. Then I have done the multiple sequence alignment tools like Gelview and Cluster Omega. These are used for the multiple sequence alignments, and then the Blast, and finally is Brenda. After these visualization tools or sequence alignment tools, I have done molecular docking with Autodoc Vina and Autodoc 4. In this, I have done rigid docking and flexible docking for some molecules. 6EQE and PET dimers, and these are the results of Autodoc 4 in rigid docking, and then the results of rigid docking in Autodoc Vina. I got some binding energy for every docking, and then I did flexible docking in Autodoc 4 and Autodoc Vina. After that, I performed the docking with canned monomer and with the ligand in Autodoc Vina, and got some binding energy and also the distance between the ligand and the enzyme. Then I did Autodoc Vina with the canned NDP monomer and the UNK ligand. Likewise, then they are given to me as assessment, and in this assessment they give the sequence of amino acid, and I have to find the enzymes and then perform the docking. And then for the identification of enzymes, I perform the Blast with the given sequences and identify the 100% match with the PDB IDs mentioned here. And then I did the Autodoc Vina with these enzymes and find the and got the binding energy. And as you see the results of my docking, the best binding energy is of enzyme 3 with the substrate. Then this is the progress month of March. In this month, I have done protein modeling with the modeler. It is used to predict and simulate the structure, behavior and interaction of the proteins, and it is used to predict the 3D structure of the target protein based on the structure of related proteins with the similar amino acid sequences. And this is the result of a modeler. I have done the modeling of canned enzyme in modeler and then did the comparative study. And then molecular dynamic simulation with the Gromex. I have did MD simulation in Gromex. Gromex is a highly versatile and widely used open source software that is designed for MD simulations. It provides the extensive tool to analyze the trajectory generated from the simulations. Or it gives the information such as structural properties, dynamic behavior, energy profiles, and interaction between the molecules. Then I have done CGNFF. It involves assigning the force field parameters to the ligand atoms and allowing it to interact appropriately with the other molecules in molecular dynamic simulations. It gives 3D structure of the ligand in the suitable format, that is STR files, and that STR file used for the further MD simulations in Gromex. Once the ligand is parameterized, that integrated into the MD simulation with the Gromex. And this is the work in progress. That MD simulation trajectory analysis, PC analysis for MD simulation, and design and calculation of MD simulation box, and understanding the various concepts of MD simulations. Then this is the future work. Bash Python trajectory analysis, plotting in GNU plot and XM Grace, then Gromex VMD TCL scripts, Solprot and Cavadoc, Plumbed and Orca. Thank you.
So just now you saw our 4 interns whom we placed in Quantumzyme present right here with what they have achieved in the last 2 months. And they are here to work for another 4 months, and possibly if the company likes them, they're going to absorb them as regular employees, not just as an intern. And I wish all of you all the best for that. One quick question to all of you is, before January, like before you joined here, you were a person, now today you are a different person. How do you see the change? Like, what is the change you see in your life?
As I told you, sir, I had a 3-4 years gap before the joining of internship. So it is a new start for me to rebuild my career. And it is really, I really got that good opportunity for restart my career.
Yeah, before joining the company as an intern, so I was searching for jobs and I was basically unemployed. So yeah. This opportunity helped me a lot to gain knowledge and gain many more skills. I'm living here outside of my home for after very long. So it's a good experience actually to learn so much and improve.
I came to this internship just after my graduation. So I wanted to know how the company works, and I had no idea where to apply, what to do, but I had said that I want to do in bioinformatics. And I saw this opportunity and I got interested in this protein engineering stuff and such. Also, I got to know how the company works. After graduation, we have no idea how the company is, how it works. So we will have, it's like a blank thing. So after coming here, we came to know how company works and such.
What is the biggest takeaway in last 2 months for you? Okay. Personally, you have grown fine, but to summarize the last 2 months of experience, what is one biggest takeaway or lesson you learned while working on job after taking the training at Biotecnika?
So I had to change my career path from nanobiotechnology to bioinformatics. So for me, the takeaway was that industrial level, there are so many things to learn and apply concepts in. So basically the application of concepts in bioinformatics aspect, I have learned them.
I have learned so much after the gap year. It is new for me, and this is a new technology and new computational tools are upcoming in this. So I have learned so much.
So that's about the takeaways. Now, one important lesson for our viewers who are watching this video, and of course all of you, is you have to know this fact that the CEO sitting in that chamber, he has to commercialize our knowledge and sell it to the world. Then only the company makes money. And only when the company makes money, it can employ more people, right? So as employees of this company or any company wherever you work in bioinformatics or biotech setup, we have to always look at how can we use this technology which we have learned during our course or during our on-job training, during our internship. How can we contribute towards the organization so that the organization can grow revenue-wise? When the organization grows revenue-wise, they will automatically come back and make you a higher designation, or they'll make you a scientist. So we all start at somewhere, right? So we all start somewhere in our lives, but later part, slowly we grow. And this is the learning curve where you all are. So I'm glad that you are making great strides. Thank you so much for sparing out time from your busy schedule as an intern in Quantumzyme. So now we will go back and see how, so the work part we have seen now, we'll see the fun part, how you enjoy and, at Quantumzyme, how is the fun factor at Quantumzyme, right? Thank you. So now we are in the last lap of our visit, and somehow I found some time with Naveen Kulkarni sir. He is the CEO of Quantumzyme and also member of Vision Group of Biotechnology Committee of Karnataka government. So we are here with him. We'll try to understand how is the dynamics of industry, what are the challenges these interns are facing, and how he is encouraging them and supporting this new generation of bioinformaticians. So let's jump in. Hello, sir. How are you?
I'm good, Shekhar. Nice meeting you, and it's a pleasure seeing that you are supporting students and their welfare by allowing, an opportunity for them to learn what happens in real time in the industry.
Thank you so much, sir. That really means a lot. So to start with, first of all, let me start by thanking you and the entire Quantumzyme team for having this agreement with Biotecnika exclusively so that we can place our students who are trained for bioinformatics with Quantumzyme. So far we have placed 5 students, and of course we interacted with them, we saw what all they learned. But now my question to you is, what's your feedback and how has been your experience with these trained bioinformaticians?
I think there is a very good plus point. The plus point is that I believe they underwent a little bit of preliminary training at Joyana
Yes.
and Biotecnika. And that sort of helped us so that we could jump straight into more and more technical aspects of the work.
Right.
And we also have a comprehensive approach in terms of how their learning path has to be. So they start with certain basics and then rigorously move on to the next phase and the phase after and so on.
Correct.
So from that point of view, one of the things that I see that's quite interesting and good is that they are very enthusiastic.
Correct.
And they're hardworking.
Right.
And they are spending a lot of time in terms of understanding what's going on.
Great.
And post that, what they read is what they are able to apply, and then again read something new and then apply that again. So these are some of the characteristics that is very essential today,
Right.
for students, because there is something that is taught in their colleges, and rarely they get an opportunity to really understand what is the use of it. Right?
Correct.
For example, quantum mechanics.
Right.
And yeah, we have all seen Marvel, and that's all we know about quantum mechanics.
Right.
Or the quantum universe.
Right.
So here we really go to that level of calculations and understand different aspects. I'll not get into the technical part of it.
Yeah.
But really some of the basics in terms of high school physics and preliminary stage college, when the basics of biology, chemistry, all of that culminate together in what we do.
Right.
And when we talk about in silico, that is in general called as bioinformatics,
Correct.
we call it in silico. In silico means computational. So the aspects of computational biology, computational chemistry, we all have heard, and I'm sure many have done some work on the drug discovery and various other areas. But what we do here at Quantumzyme, that is something very unique. That is applying quantum mechanics for enzymes.
Right.
That is in a direction or in an area where we want to be more, let's say, climate aware and environmentally aware, and contribute positively to Mother Earth, if you say so, to be able to provide a greener solution for chemical manufacture.
Right.
Now, all of this, how do you put together? And that's where all the big challenge is. Now, if I expect some fresh student to already know and understand and then be ready to be productive, it's very, very difficult.
True.
And what I think together with Biotecnika we have accomplished, that I'm very happy to say, is that the whole industrial training, like a live project working and applying the basics of what they have learned
Right.
in real time to solve specific problems.
Right.
So that is something that's a rare opportunity for them. But also for us, I love working with students. They are filled with energy.
Right.
They are enthusiastic. They're always eager to go and do. In fact, they're doing something very, interesting, because there is one of the software that we found it very difficult to install, and people did that time and all of that.
Right.
And you know what they did? They just went to ChatGPT or such tools, and they started getting a lot of answers, and they've started trying. And I'm being told that by this weekend they'll be ready to install, and it will be installed rather than ready to use.
Wonderful.
So these kind of problem-solving abilities, you will find it only in real world.
Correct.
In a regular lab, everything is set and they just go and start doing their things. It's like, my laptop or my mobile loaded with all the apps that are required and I start using it.
Correct.
But here we are developing the app. And
Right.
that's the difference that brings it. And I really love working with students. And one of the important things that we have today is in the government of Karnataka, there is a separate committee that has been established for skill development.
Yes.
It's part of the Karnataka biotech policy to support employability of students. Develop skills that makes them more and more employable by corporates or companies.
True.
In fact, that's what we need. It's all about translation now,
Right.
from learn to apply. And that is something that's a gap that together with the industry, we are working and trying to support that and bring that the workforce that is there in India
Correct.
to be able to even deliver.
Now that we completely understand what Quantumzyme is doing, and I'm happy that our students are able to contribute to the growth of Quantumzyme as interns. So my next question obviously would be, would you allow us to place more students with Quantumzyme?
Okay. Honestly, I would have asked that question because I feel there is a tremendous force that can be created. There is a lot of advantage and benefit which is mutual in nature. Now I'm talking about students and Quantumzyme,
Right.
Biotecnika aside,
Right.
because already you're doing a great service working with hundreds and thousands of students. For us also, a few benefits that we see is partial training already provided, and then they come here, and then we have our learning path. Where for the first 2 to 3 months they learn a lot, and our scientists give them tasks, give them small tasks and bigger tasks and bigger and bigger. In fact, last week they made a presentation on a very interesting publication, and the first draft was not at all good. And once they got the feedback, they worked throughout the weekend. Friday was a Good Friday holiday,
Right.
and they worked throughout Friday, Saturday, Sunday, and Monday they made a presentation which was really out of the world.
Wonderful.
And when we saw that, we were also able to see a lot of interesting insights
True.
that they were able to bring out. And many times we don't have time to go through all these interesting publications.
Correct.
But with these kind of interactions, we also learn it's the energy, the passion that they bring in. So definitely, Shekhar, we will be very, very happy to have more. One of the things is that many times students become a little, lethargic, or they are not serious and so on and so forth. So there has to be a way we can get a bigger,
Serious
better commitment. Yes.
Yes.
So that they are committed. The reason also is our scientists spend time in training them and spending time with them.
Correct.
And teaching them and all of that. And by the end of the day, maybe for the first few months, it may look like some hard work from our end as well. But after that, once they become productive, once they learn, they can start doing some small tasks and small things and start helping and supporting us. So that's a very good synergy that we can develop.
Right.
And I think already in the first batch, you have done a very good screening, and we've got very good response.
Thank you. Thank you so much.
And great, I would say, talent pool is out there,
Right.
waiting to be, I think, explored and to explore, if I may say. And that's something that we should leverage.
Right.
And happy to see what more we can do, how else we can work better. And please bring us more.
Wonderful.
And happy to work with many.
Great. That's really awesome. So you heard Naveen Kulkarni sir, he is ready to have more interns in his company. This is a partnership between Biotecnika and Quantumzyme, where we train and we place you as an intern in Quantumzyme, where you work on real world quantum chemistry, green chemistry, quantum mechanics, and bioinformatics. So whatever you learned with Biotecnika, you can apply in the real industry, and you get to work with the best of the scientists in our country, and they are working with Quantumzyme. So what are you waiting for? You can enroll with Biotechnika's Bioinformatics Guaranteed Placement with Quantumzyme program. The program link is given below this video. And if you have any questions or comments, please feel free to comment below or reach out to me at shekhar@biotecnika.org. All the best, and I'll see you soon as an intern in Quantumzyme. Thank you. Take care. Bye-bye.